Topic 3, Virus Quantification. This content is part of the module MBS 7117, Molecular Microbiotechnology. It is housed in the Department of Biomolecular Laboratory Technology and Molecular Biology, abbreviated as BLT, in the School of Biosecurity, Biotechnology and Laboratory Sciences, SBLS, in the College of Veterinary Medicine, Animal Resources and Biosecurity, COVAB, Makerere University. I am the presenter for today, Dr. Penny Nansamba. Our content on topic 3 consists of number 1, definition and justification for virus quantification, types and assays for quantification of viruses, Plaque titration assays, and lastly, number 4, infectious dose 50. So this is the second series in topic 4, and we are going to concentrate on number 3, plaque titration assays. The learning objectives for topic 3 are as follows. By the end of this lesson, you'll be able to, number 1, justify virus quantification. Number 2, list methods for virus quantification. Number 3, explain the plaque titration. Number 4, explain the infectious dose 50. In this second series of topic 3, we are going to concentrate on objective 3. Explain the plaque titration. So here we are in the second series on topic number 3, and we are concentrating on plaque titration assays. Learning objective 3. By the end of this section, you'll be able to explain the plaque titration, highlighting the principle underlying it. So let's give an overview on the plaque titration assays, and we are going to start with a definition. A titration, or different concentrations of the virus, is applied to a constant number of cells in culture, and the virus is given time to replicate. Let's depict this in a diagram. We have a stock virus in the conical flask, and now we're going to make different concentrations of the virus. So we are going to titrate the virus into different concentrations. We usually use 10 to the minus 1, 10 to the minus 2, minus 3, minus 4, and so on, because 10 is an easy number to deal with. So once we have diluted our virus into different concentrations, or titrated our virus into different concentrations, we are now going to apply them to a constant number of cells in culture. We are going to grow monolayers, perhaps on a multi-well plate. It may be a 6-well plate, 8-well plate, depending on what you have at hand. Now we get our multi-well plates, and perhaps to the first row of wells, we apply virus at 10 to the minus 1. Then to the second row of wells, we apply virus at 10 to the minus 2. To the third row, we apply virus at 10 to the minus 3, and so on, until we've applied all our viruses up, our different concentrations of virus, up to 10 to the minus 8 or minus 9. So note, you may have to have multiple plates, not only one plate. So all of this would be day 1. After applying the virus, it is given time to invade and replicate. And after some time, this is what we are going to see. This would be perhaps around day 5, where we note that the application of virus to the cells results in plaque formation, and we can quantify the plaques. So back to our definition. Infectivity is then quantified using the plaque titration assay. It's noted that the plaque titration assay is common and it's a gold standard. Now let's look at the procedure used for a plaque titration assay. Number 1, we need a confluent mono cell cultures. So we obtain multiple wells and we prepare monocultures on these multiple well plates. We usually prefer a confluency of about 80% to 90% because as we incubate for several days, our monolayers continue growing and we don't want them to stop growing or to begin peeling off. So we look for a cell line that is permissible to the virus we are going to quantify. The picture below shows 6 well plates. It's only one plate, and in the 6 well plate we have a monolayer of confluent cells up to 80% confluency. Now let's go to step 2. We are going to prepare serial dilutions of viruses, or we're going to titrate our virus. We usually use 10-fold dilutions because it's easier for subsequent calculations. And we're going to dilute our virus to 10 to the minus 2, minus 3, minus 4, and you can even go up to 10 to the minus 8 or 9. So how do we do this? We have our stock virus, which is in the first tube in the picture depicted below. So with this stock virus, we are going to get an empty tube and fill it with 990 microliters of culture media. Then we are going to get 10 microliters of our stock virus and put it in this tube that has got 990 microliters of culture media. Discard the pipette tip that you're using on the Eppendorf, put in another, and agitate it gently to make sure that all the virus has mixed with the culture media and we've got a homogeneous solution. So there we've got our virus that has been diluted to a concentration of 10 to the minus 2. From this we're going to obtain a dilution of 10 to the minus 3. So again we get an empty tube and fill it with 900 microliters of culture media. So there we are, we have 900 microliters of culture media in this tube. We get our virus that has been diluted to 10 to the minus 2. Pick 100 microliters of this diluted virus and add it to the 900 microliters of culture media. And we've got a dilution now of 10 to the 3. Homogenize the mixture. Get another tube, fill it with 900 microliters of culture media. Now we're going to make a dilution of 10 to the minus 3. Get our tube at 10 to the minus 2. Obtain 100 microliters. Put it in the 900 microliters of culture media. And we've got our dilution at 10 to the minus 3. And so on. So once we're done with the dilutions, we can move to step number 3. We are going to titrate our virus onto our confluent monocell layers. So where are they? Okay, there they are. We are going to maintain a constant volume of virus onto our confluent monolayers, but this virus is at different dilutions. So we get our virus at 10 to the minus 1 or 10 to the minus 2, get 10 mils of this diluted virus at 10 to the minus 2, put it in the first row of our 6 well plates. Get 10 microliters of our virus diluted at 10 to the minus 3, put it in the second row on our 6 well plates. Get 10 microliters once again of our virus diluted at 10 to the 4, put it in the third row of our 6 well plates. And we continue doing that. Note that you may have to have several multi-well plates if you're going to continue the titration up to 10 to the minus 8 or 9. So now we wait for the virus to infect the cells, can give it about 1 hour. And then we are going to cover our monolayers with an immobilizing overlay medium to prevent viral spread of CPE. Now let's look at the principle underlying this. Once an infectious virion infects one cell, the progeny that are produced can only infect an adjacent cell because of the gel overlay that restricts movement. Remember, it's an immobilizing overlay medium. So after some time, we are going to get a localized area of infection called a plaque. Now let's go to step number 4. We allow the plaques to grow until they're visible. So this is what we're going to see. And then, let's look at these plaques once again. With our virus at a concentration of 10 to the minus 5, we have many more plaques. When we dilute our virus to 10 to the minus 6, there's fewer virion particles there, so we have less plaques. When we dilute it at 10 to the minus 7, we even have much more less plaques. So when our plaques are visible, we are going to now fix this monolayer with formaldehyde, and then we are going to stain it with a dye for contrast. Methyl blue is preferred, or we can use crystal violet, and then we count the plaques. Let's look at the principle underlying. The formaldehyde, which we use, it fixes the cells, maintaining the structure so that they do not wash off the plates, and it also kills the virus, making the whole assay less infectious. Then the stain provides a contrast where we can differentiate areas of CPE, these are the plaques, with areas where there are no virus infection. So this would be actually taking up the dye. Now we are able to calculate the titers in terms of plaque forming units. The stock titer can be calculated based on the following. We count the number of plaques. We note the sample dilution at which the aliquot was drawn for the count. And then we also take note of the volume of the aliquot that was added to the cells. Now let's look at this in a pictorial form. These are the plaques that we were seeing. If you look at a dilution of 10 to the 5, those are too many plaques to be counted. So we can't take that dilution. If you look at 10 to the power 6, those plaques are well spread and they can be counted. They are ideal for counting. A total of 16 were counted. If we look at 10 to the minus 7, those are too few plaques. Even if they're 4, they're too few plaques and they'll give us an error. So we've counted 16 plaques, and these were at a dilution of 10 to the minus 6. And if we can remember, we were using a volume of 1 ml of aliquot to put on our 6 well plates. So that's the volume of aliquot we added to the cells, and it was uniform all through. So now we can refer to the formula. The virus titer in plaque forming units, or PFU per mil, is equal to the average number of plaques divided by the dilution, whether it's 10 to the 5, 10 to the 6, or 10 to the 7, multiplied by the volume of aliquot put on the wells. Here it's going to be the 16 plaques counted divided by the dilution, 10 to the minus 6, because that's the dilution at which we counted the plaques, times 1 mil. That was the volume of aliquot that we added to the wells or the cells. Now, what are the strengths and weaknesses of the plaque assays? Let's look at the advantages. It's a quantitative assay, which is good, and it only measures infectious particles. Disadvantages: it's lengthy and time consuming. It's going to take you about 1 whole week before you can get results. You'll have spent 1 or 2 days getting the monolayers on the multi-well plates. Then you titrate on the virus on the 2nd or the 3rd day. Leave the plaques to develop for about 3 to 5 days. No, it's taking no less than 1 week. And it's only suitable for viruses that show CPE. Now, I would like you to look at this short video that depicts the plaque titration assay. It's about 8 minutes long, but it will help you appreciate the assay. It's at the URL given below. So in summary, the lessons learned so far: we've looked at the procedure for carrying out the plaque titration assay and the underlying principles. The next step: please refer to the lecture notes given to you on the study portal, the activities therein, the chapters in the textbooks, and the peer-reviewed journals that we have recommended you for further reading. This has been our source of information. Thank you.